Hello everyone, welcome to BioInsight Academy by Yasmin and Sarah. In our first video, we're going to talk about the CRISPR-Cas genome editing. So let's get it started. Alright, we may know that CRISPR-Cas editing technology has numerous applications across the various fields, such as medicine and therapeutics, gene therapy, cancer treatment, infectious diseases, and even in agriculture. So based on our goals and targets, different Cas protein can be used. So at first, let's start with Cas9 protein. As you can see here, there is a Cas9 protein and double-strand DNA. Cas9 protein recognizes a short DNA sequence known as PAM for the commonly used Cas9 from the Streptococcus pyogenes, SP-Cas9. The PAM sequence is NGG, where N can be any nucleotide. The presence of PAM sequence adjacent to the target DNA site is crucial for Cas9 to bind and initiate the cleavage. As you can see here, there is the sgRNA. This engineered RNA molecule combines the functions of the naturally occurring CRISPR RNA and transactivating cRNA. Totally, sgRNA has two main components: spacer sequence, approximately 20 nucleotides long, complementary to the target DNA sequence, and the scaffold sequence helps in the formation of a complex with Cas9. The sgRNA guides the Cas9 protein to the specific target site in the genome by base pairing with target DNA sequence. Once the Cas9 sgRNA complex locates the target DNA sequence adjacent to the PAM, Cas9 induces a double-strand break in the DNA at a specific location within the target sequence. Actually, Cas9 has two nuclease domains, RU with C, which code the complementary, and H and H, which cuts non-complementary strands of the DNA, resulting in DSB or double-strand break. After the double-strand break is created, the cell natural DNA repair mechanisms are activated to repair the break. There are two primary pathways for repair. One of them is NHEJ, which is non-homologous end joining. This is an error-prone repair process where the broken DNA ends are directly ligated without the need for a homologous template. NHEJ often results in insertion or deletions or indels at the break sites, which can disrupt the gene and lead to loss of function or knockout. However, there are another mechanisms called HDR, which is homology-directed repair. This is a precise repair process that uses a homologous DNA template to repair the break. Actually, researchers can introduce a repair template with the desired sequence flanked by homology arms that match the sequences adjacent to the DSB. HDR can be used to introduce the specific mutations, insertions, or corrections, or knock-in. Well, the second CRISPR-Cas system which can be used in genome editing is Cas12a, which is known as CPF1, is another CRISPR-associated endonuclease that has a distinct properties and mechanisms compared to Cas9. Cas12a actually recognizes a different PAM sequence compared to Cas9. For Cas12a, the PAM sequence is typically TTTV, where V can be A, C, or G. This Cas12a utilizes only a cRNA to guide it to the target DNA sequence, which consists of direct repeats, which are a short sequence that is part of the cRNA and helps in the formation of a complex with Cas12a, and the spacer sequence, approximately 20 nucleotides long, complementary to the target DNA sequence. But unlike Cas9, Cas12a does not require separate trans-activating cRNA for its functions. Well, Cas12a cleaves the DNA in staggered manner, creating the sticky ends or overhangs rather than the bond ends produced by Cas9, as we said before. The ROFC domain of Cas12a is responsible for the cleavage. It first makes a cut in one strand of the DNA, followed by a cut in the second strand, resulting in a double strand break with sticky ends. After the DSB or double strand break with sticky end is created, the cell's natural DNA repair mechanisms are activated to repair the break, just like Cas12, as we said before. So we said that there are two DNA repair mechanisms, NHEJ and HDR. Next is Casa30A, which is another member of a CRISPR family, is distinct from Cas9 and Cas12a in that it targets RNA rather than DNA. Casa30A uses a CRISPR RNA, cRNA, to guide it to the target RNA sequence, and it consists of a direct repeat, a sequence that helps in the formation of a complex with Cas30A, and spacer sequence approximately 20 to 30 nucleotides long, complementary to the target RNA sequence. The cRNA binds to the Cas30A, forming a complex that can recognize and bind to a complementary RNA sequence within a cell. Once the Cas30A cRNA complex binds to the target RNA, Cas30A cleaves the RNA, unlike the Cas9 and Cas12a, which cleave DNA. Cas30A specifically cleaves RNA. So Cas30A not only cleaves the target RNA, but also exhibits collateral cleavage activity where it can cleave another nearby RNA molecules non-specifically once activated. This property can be harnessed for various applications, including diagnostic assays. Another member of a CRISPR-Cas family is Cas40a, which is distinct in its ability to target and cleave single-strand DNA without the need for a PAM. So Cas40a uses a sgRNA to direct it to the target ssDNA, and it consists of a spacer sequence approximately 20 nucleotide long and a scaffold sequence. So the sgRNA guides the Cas40a protein to the specific target ssDNA by base pairing with it, and it cleaves the ssDNA, resulting in a single strand break rather than having a double strand break that we had in Cas9 and Cas12a. So this activity is highly specific to ssDNA and does not require the presence of a PAM sequence, unlike other CRISPR-Cas systems that we talk about. Well, next is CBEs, or cytosine base editors. Type of CRISPR-Cas system that allows for precise single base pair changes in the DNA sequence without generating double-strand breaks. The single guide RNA, or sgRNA, directs the base editor to a specific locus in a genome by complementary base pairing with a target DNA sequence. The Cas protein, as you can see here, used in the CBEs is either a dead Cas9 or nickase Cas9. So this version of Cas9 can bind to DNA and induce a single strand nick, but does not generate DSBs. The Cas9 or nickase Cas9 is fused to the cytidine deaminase enzyme. For example, such as ApoBEC1. This enzyme actually catalyzes the amination of cytosine to uracil, so C2U conversion. So the cytidine deaminase converts a cytosine base in a target DNA strand to uracil. During the DNA replication, or repair, the cell's machinery recognizes the uracil and replace it with the thymine, so U is read as T, resulting in a C2T and G2A on the complementary strand base change. So, in this system, for generating staggered cut and single base replacements, first the Cas9 nickase creates a single strand nick is in the non-edited strand opposite the deaminated cytosine. This nicking helps in stimulating the DNA repair process, enhancing the efficiency of base editing. And finally, the last CRISPR-Cas system which can be used in genome editing and we want to talk about today is ABEs, or adenine base editors, which are designed for precise single base pair changes in a DNA sequence, specifically converting adenine to guanine, which result in corresponding thymine to cytosine change on the complementary strands. So here again, the single guide RNA directs the base editor to a specific locus in genome by complementary base pairing with a target DNA sequence. And similar to CBEs, as we said, the Cas protein used in the ABEs is either a catalytically dead Cas9 or nickase Cas9, which this version of Cas9 can bind to the DNA and induce a single strand nick, but again, as was said, in CBEs does not generate DSBs or double strand break. So the Cas9 or Cas9 nickase is actually fused to an engineered adenine deaminase such as TADA or TADA, which catalyzes the deamination of adenine to inosine. Inosine is then interpreted as a guanine during the DNA replication or repair. The adenine deaminase converts an adenine base in the target DNA strand to inosine. So during a DNA replication or repair, the cell's machinery recognizes inosine as guanine, resulting in an A to G and T to C and the complementary strand base change. If the target site initially had an AT pair, it will be converted to GC pair. So the process involved targeting a single locus, creating a staggered cut or nick, and achieving the desired base substitution. The Cas9 nickase creates a single strand nick and a non-edited strand opposite the deaminated adenine. And this nicking helps to stimulate the DNA repair process, enhancing the efficiency of base editing. So today in this video, we'll learn about the different CRISPR-Cas systems which can be used in genome editing based on various targets and goals. Thank you for watching our video and being with us in BioInsight Academy by Yasmin and Sarah. If you enjoyed the content, please subscribe to our channel, like this video, and share it. We appreciate your support. Please don't hesitate to leave a comment below if you have any suggestions or ideas to help us improve our videos. Actually, your feedback is valuable to us. Once again, thank you for watching, and we look forward to sharing more content with you soon.